And thanks a lot to Francesco for letting me borrow, having a laser pointer, and the Illumina representative for the same thing. Let me first start with the acknowledgements. Now I'm an assistant professor at Hopkins, and this is really the work of a lot of people in the sort of methylation community in Hopkins. Primary Raphael Irisari and Andrew Jaffe has contributed a lot, but on these slides here, there's ideas and work from all the people here. Martin Arai, who is now at Mass Gen, Hector Corrado, who has his own lab down at UMD College Park, Chris Adacosta, Danny Fallon, and of course Andrew, and his group for sort of discussion about data issues. And we are always looking for postdocs or grad students. I think most people here are probably at the grad student level, so it's not so relevant. But if you want to get to Hopkins, send us an email, and we'll see what we can facilitate. OK, so I'm going to talk about Minfi. And Minfi is really a bioconductor package for the analysis of 450K data. And really, the way we think of Minfi is that this is a package we primarily made for ourselves and for other developers. And it contains sort of data structures for representing 450K data at various stages of an analysis pipeline. And we think this provides a very flexible and clean development framework for implementing and developing new methods. We are very open to including code from other people, methods that have been developed or can be fit into our development framework, and hosted in the package if you don't really want to maintain the stuff. And one example is Swan, that a lot of people have heard about. And while we kind of know the Swan developers, and we have beer with them once in a while when they get to the US, this is not really our work. They have like their own method, and they have their own sort of part of the package they take care of. If you want to contribute something, we really want some man pages, because that's the worst thing about writing code, and preferably some clean code and some unit testing. OK, so Minfi has a whole slew of classes. I just want to present it very briefly. The sort of most fundamental of the first class represents the red and the green color channel for all the probes in the array. And it's really something you create from reading an IDAT file. The next class, which where you can make where you do some kind of pre-processing to, is you get an object that represents the methylated and the unmethylated channels on the array. And this is a non-invertible transformation, because there are probes that we're going to hear about later on from Kim Sigmund, which are kind of discarded when you make this transformation here, because they don't really represent methylation. Then you can take your methylation, you can map it to the genome, and again, that's sort of non-invertible, because you have probes that don't map to the genome. Not a lot of them, but there are a few, and you get rid of those. And then you can imagine taking your methylation, non-methylation channel and converting that into some log ratios or some beta values, possibly supplemented with copy number. And because we have all of these classes here, you can have pre-processing methods that go like directly from the red and green channels directly down to the genomic ratio set if you want to do some magic stuff on the way. We have a pre-processing algorithm that we haven't really published or made publicly available yet, but that does this stuff here. OK, so we have a couple of new functions that are still not in Minfi, and would have been nice if I was here and gave my talk and I was like, everything is in there, you can just download and use it. But I have a newborn, and that has slowed down process significantly. But we have some things that are pretty simple, getting some data out that has been a little bit too hard to get hold of. Some quality control. We have talked about estimating this cell composition, which I'm going to talk about at length. We have our pre-processing method. We want an interface, an easy-to-use interface to SVA, which is a method for correcting for batch effects. And then we have two functions for finding small and large-scale changes in the epigenome. And all the stuff up here is pretty close to being released, and this stuff down here is a little bit further away. So this is very much work in progress. So when you do an analysis, I just want to mention this very briefly. You have a lot of samples. You always want to do some quality control, and it's always a little bit hard to know how do I do quality control, and there's various ways. This is something we like. It's very, very simple. You take each dot here is an array. You take the methylated channel and the unmethylated channel. You calculate the overall median of all your loci. So 2 numbers, you plot them against each other, and you see that most of the data is up here, where you would expect it. And then you have some arrays down here, and we would posit that these arrays here are generally bad and should be removed. We have some debate on whether the cutoff should be at 10.5 or 11 or perhaps 11.5. But there's definitely some funky stuff going on with the arrays down here. So I'm going to show some plots of the beta values for an array up here, an array here, and an array here. And like I said, this is just median overall intensity. So this is something you would expect to see. You have this nice bimodal distribution of, sorry, it's not the beta values, it's the M values. As you get further down, the 2 peaks move closer together. And when you're sort of all the way down at the bottom, you have like 1 peak. So this is clearly a screwed up array unless you are doing something really funky with your samples. So this is quite easy to do. It takes like 5 minutes. And we have found it to be pretty effective. Now, when you're doing a large-scale study, you should always think about sample mix-ups. There are sample mix-ups in essentially all large-scale studies, and there's a paper, I forgot which one, that claims that up to 10% of all samples in any GWAS study has been mixed up, where you have the wrong labels to the wrong chips. So this is potentially a pretty big issue. You should always, it's not sexy to talk about, but you should always spend a lot of time thinking about it, because it can kind of impact your analysis. And on the 450K array, we have some SNP probes that are kind of meant to look at this. But one thing we have found sort of a little bit useful, which is, again, very easy to do. You can predict the sex of the sample from the intensities on the X and the Y chromosome. This is pretty easy to do. You get like, you can quite easily see which are males and which are females. You can compare that to the reported sex of the samples, and then if it doesn't match up, you typically have a problem, or you have somebody who went through a gender change. But this is, again, very easy to do and helps a little bit. So it's not sexy to talk about, but spend some time worrying about it when you do an analysis, especially when you have a lot of samples. Now, there's been some talk about cell type composition today, and I'm going to show some reason. The mics are up here, so I shouldn't go too far away. I'm going to show a little bit of recent work from our group on this, but essentially the opposite is this. The epigenome varies from cell type to cell type. And the blood, the tissue that most people use for doing EVOS type studies is blood, and that's purely for convenience. Like Christoph totally correctly said, blood is a terrible tissue for most diseases we're interested in, but that's what people have in the freezer. And it's like one of the only tissues you can get hold of pretty easily from people. I mean, it's hard to get brain samples. People don't like to give that up. Now, blood is composed of a lot of different cell types. There are many cell types, but there are sort of 6, or we tend to think of 6 primary cell types. And in a seminal paper that I really think everybody doing EVOS should read, Hausmann showed that this can or will confound studies of DNA methylation performed on blood samples. He did this on a 27K array, and he used the fact that you can flow sort blood. So you sort it out into clean cell populations. You estimate something you think of as the cell type specific methylation profile, and you use that to estimate the cell type composition. I'm going to show this. This requires flow cell sorted blood, and in a paper on PLOS Run, Reynolds did this exact experiment on the 450K array data. So we can download that and play with that. There's only 6 samples, but 6 samples is better than none. And obviously, this can be a problem for other tissues. There's a recent paper in epigenetics where they studied 2 different cell types in brain and showed that it can also confound the analysis in brain. This is a really, really big headache for EVOS type studies. I mean, everybody uses blood, and everybody has to think about this. So I'm going to show like an example of possible effects this has. Now, Hausmann had a nice algorithm, and essentially he selected a small set of markers on the 27K array that he said, I can use these markers to estimate the cell type composition. I want sort of fairly clean markers that very clearly identify the cell type of origin. So these markers are largely also on the 450K array, but in our hands at least, we didn't find that these markers performed as well on the 450K array as it did on the 27K array. So for the plots where I have 450K array data here, we kind of did the same thing. It's essentially the exact same algorithm, but we used different markers. We went through kind of the marker selection step and all that stuff. And while it's not in Minfi yet, it's going to be in Minfi really soon, so you can kind of do it yourself. Okay, so aging has been studied a lot in DNA methylation, and this is data from 6 big papers with a lot of samples removed because we think they're bad. And up here we have the estimated proportion of monocytes in the sample. So you have essentially 6 plots like this. And one problem you have with studying this in aging is that the study and the sample age is pretty confounded. So the people who study young people, and the people who study old people. So there could be all kind of batch effects and so on, but I'm going to show a little bit later that there's probably a problem here. So the issue here is that the proportion of monocytes changes throughout time. And this makes biological sense. We actually know that certain blood cells are generated in smaller and higher proportion as you age. So the issue here is that if you see a difference in methylation between, let's say, newborn and 80-year-old, you better make sure that it's actually a difference in methylation and not just a difference because newborns have a different blood composition. Now, the fact that newborns may have a different blood composition than old people is a biological finding, but it doesn't really tell you that methylation or age is encoded in a molecular marker in the genome. Now, what we have here is data from the 450K and the cell-sorted stuff. Here we have different blood cells, and we have selected a number of probes here for like optimizing differences between the different cell types. And you can see here very clearly that you have some very clear clusters, or the different cell types correspond to very clear clusters. And on the next page, I'm going to concentrate on only 2 cell types. So this here is going to be zoomed up now as here. So here we have some probes, and we have two different cell types, and there's like a huge difference here. Now over here we have real data, or this is real data, but this is also real data from young people and old people. And you can see there's a huge difference here that largely visually corresponds to this thing here. So what you do is you use these mentioned loci to estimate the cell proportions, and you get this stuff here down here, where you can see that here on these samples here we have a lot of the granular sites, and here we have a sort of a more even mix. And down here we are forming sort of this linear combination. You can see here's the estimates for this sample here and for this sample here, which are kind of cut out here. And the difference here is that we estimate that the granular size goes from 28% to 75%. And that essentially drives, we will claim, the major difference on these probes here. But this is just a small set of the probes in the array. So you can take the data and you can do a principal component analysis to figure out what drives the variation in the data. And up here we have the first PC, the most variable kind of direction in the methylation space. And down here, we just take the cell counts. There's 6 different cell types. They sum to 1. We do a PCA. That's essentially we sum up the cell heterogeneity in one number, and we can see there's a huge correlation, or a pretty good correlation, between the first PC of the methylation data and the first PC of the cell type counts. This very strongly shows that blood composition is a major driver in the variability in this data here. So just to be safe, all of this data here was sort of downloaded and renormalized. Now, this doesn't tell you that the findings they have in these papers are necessarily false, but it does show that there's a major impact of age on age and cell type composition, and that is likely to explain a major component of the variability. So in the most extreme case, we have the yellow stuff here, where we have, they reported roughly 2,000 loci that they said were associated with AIDS. And we take those 2,000 loci and we correlate the methylation level at these 2,000 loci with our blood composition. We have 6 correlations because we have 6 different cell type 6 different cell types. We take the maximum of those correlations. And you can see that a lot of them are quite heavily correlated with a major component with one of the cell type compositions, suggesting that a lot of these things here are probably not really associated, are not really age-driven markers in the epigenome, but rather driven by differential cell type composition. Although there's stuff down here that very well may be true. But there's a difference between saying 2,000 markers are associated and let's say 20, 50 or something like that. This is from a different study where they did it on the 450K array. They actually reported 70,000 CBGs being associated. There's only 80 here because that was what they had in the supplementary material. We didn't really want to redo the analysis. But there's still some major correlation here, and perhaps they selected these 80 here for sort of various reasons. But anyway, this is a really big issue, like Christoph is saying and other people have been saying. Reviewers are really gunning for this, and if I'm reviewing papers, I'm gunning for it. So people who do large-scale studies in blood really have to address this. And the first thing you should address is, could this potentially confound your study, right? The really big issue is if you're studying diseased people and non-diseased people, and the diseased people have a different cell type composition from the non-diseased people. That is, in some way, most likely to occur in diseases that had a direct effect on blood, but those are also the diseases that are most likely to be detected when we use blood as tissue samples. Now, you run this stuff here, you get some cell type composition estimates, and what do you do with it? Well, that is, I think, still a little bit of an open question. The standard thing is you take these 6 observations, let's sum to 1, you perhaps do a PC on them or something like that, and you regress out the signal and analyze the residuals. That has been reasonably popular. We are not really sure that that is so good. These cell type compositions, the numbers, the vector we're estimating down here, has quite a lot of variance associated with it. And when you do this regression trick here, you're really assuming that this here is estimated without error. And so that makes it a little bit dangerous to just put it into a linear model. So I think it's still an open question what you do if you're really affected by it, but what everybody can do is at least have a pretty strong indication of whether their findings are possibly confounded by this issue. Okay, so Christoph and Tiffany discussed batch removal. I don't have a slide on it, but if people in this audience are doing EWAS type studies and don't know about correcting for patch effects, they should learn about it. We have found that using SVA, assuming that cell type composition is not correlated with your variable of interest, that SVA to some extent removes the effect of cell type composition, as you would kind of expect if you think about it a little bit. SVA is a method for automatically removing batch effects that we like. Okay, so now I'm going to switch over to talking a little bit about DNA methylation, detecting differentially methylated regions. Okay, so on the 450K array, what comes out of your data is really single CBG measurements. The first couple of slides illustrates possible pitfalls of trusting single CBG methylation measurements, although I have to say that this is not data from the 450K array, so it kind of doesn't really support my point as good as it could. But here we have 2 different CBGs. You have a huge difference in cases and controls, and you would say, hey, this methylation difference here really drives, there's a huge difference here. And over here, you have a difference, but it's not so good. So you're thinking like, which one of these two loci do I trust more? And if you do like a t-test, this here will definitely come up as being more significant, I would say. Now, if you look at it in sort of a high resolution sense, this is the first CBG. It's in this little thing here. And you can see, the curves here are smoothed methylation estimates. They're kind of regional level methylation estimates, pooling information from sort of nearby CBGs. And there seems to be very, very little happening here. And you have to stop and ask, well, is this really a true finding or is it a false finding? And that you can contrast with a picture like this, where you have tons of nearby CBGs that act together. Now, this is not from the 450K array. This is from an assay called JARM, which is a restriction enzyme-based assay. It doesn't have the same resolution. You can definitely argue that JARM is more likely to have these issues than the 450K array. But we still believe that you have to think a little bit about single CBG measurements. There's also some biology. Recently, we and other people, there's a slide with other data, profiled. This is cancer normal. The red ones are cancer. The blue ones are normals. Profiled cancer using whole genome bisulfite sequencing. And what we often see is kind of these more region-level shifts, where here you have, for example, in the normals, this is around the CBG island. You have a very tight boundary with high methylation outside of the island and low methylation in the island. And in the cancer, this structure seems to be completely erased. Down here, you have a case where in the normals, you have like a tight boundary. And you kind of have a tight boundary in the cancers as well. But in the cancers, the boundary kind of expands out, and the island kind of gets bigger. And when you are analyzing a single CBG, you kind of make a line here and you say, this is how it happens in this island. And if you had placed your CBG here, which you would do if you used the 450K array, you wouldn't really see these things unless you had a probe here. And if you had a probe here, it kind of mattered whether the probe was here or here, and so on and so forth. You get the point. This is not just in cancer. This is data from a recent paper from Greg Hannon's lab, where they also did whole genome bisulfite sequencing on different tissues. And here we have kind of a DMR, also near an island, but not really the island. I mean, it's sort of close to the island. And you kind of see the same thing here, right? You have kind of a region of low methylation, where the width of the region kind of changes in different tissues. Rather interestingly, they actually say in this paper, as far as I can recall, and I don't think they have a whole lot of data to show for it, but it's pretty interesting, that the relationship with gene expression is strongest if this expansion happens in the direction of the gene body. But I think we need to confirm that. Okay, so let's take the 450K array, and I've downloaded some samples from GCGA. I'm just analyzing the normal samples. So this is really noisy data. I'm not doing anything. I'm not removing SNPs. I'm not doing batch correction. I'm not doing anything. I'm just taking the M values, and I do a t-test on the M. I do some statistical test on the M values. I correct for multiple testing using Benjamini-Hochberg, which controls the false discovery rate, and then I only look at CBGs that are significant and that are covering that are 2 kb within the transcription start site of a gene. And up here on the x-axis, I have an expression measure on the nearby gene. And this is barcode normalized data. This is on the log2 scale, so it's a pretty normal scale. Down here, I have methylation differences on the beta scale. So these were all significant. I have to say that because many genes have multiple probes in or near the promoter of a gene, there are definitely multiple points here for each gene. They are not, so there's about 70,000 points here. And clearly, this picture here shows that this analysis here is really not what you want to do, right? First off, it's pretty interesting that you have a huge number of points where the actual effect size on the beta scale is like 0. And I see that in the literature sometimes with EVOS data as well. You report effect sizes that are very, very small. And I have to say, a lot of us are interested in DNA methylation, but for a lot of diseases, a lot of like systems, except perhaps for sort of the big ones like cancer and cell type differences, tissue differences. We don't really know, even if we know that there must be an epigenetic mechanism in the disease, we don't really know that DNA methylation is related. Reporting loci with a beta value that is this small here is like, in my opinion, we're not really ready for that yet. Even if you have a change of 20% on the beta value, we have for many methylation loci in the human genome, we don't really know the functional impact of it. We don't even know it does anything. Reporting something that has a minor difference is pushing it too long. So you can you can lower your p-value threshold, and I think you can do that in many ways. Here I've just done it by using Bonferroni instead of Benjamini-Hochberg, so now I control a different error rate, blah, blah, blah. And we remove like tons of points in here, like 40,000, and now we are beginning to see sort of a separation between the cloud on the left and the cloud on the right. The red lines that are perhaps a little bit hard to see here are plus minus 5% and plus minus 10%. And while I have no support for choosing it, you have to be pretty convincing to make me believe anything less than, I don't believe anything less than 5%. 10% is still kind of a little bit of a stretch, I would say. So what can we do about this? Well, we can do kind of the same approach as we've been using on whole genome bisulfite and JARM and other analysis, and that's essentially use the fact that DNA methylation is spatially correlated and pool information on nearby CBGs. So there are roughly 115,000 probes on the 450 array that are isolated. And by isolated, I just mean they have a distance that's greater than 1 kb to the nearest other probe. Well, when I say probe here, I should really say methylation loci. So these ones here, we can argue we perhaps can't get very much information out of pooling it, but there's 300,000 probes that are pretty close to a lot of other probes. Right? That's still a lot of information that's left. And up here you have a picture. This is a colon and lung, or perhaps it's lung and colon, I forgot. And you can see here that, let's see, this is a 3 kb region. And over this 3 kb region, we have tons and tons of probes here that are all highly different between the two tissue types. And this is, I would argue, when you see a picture like this, it's much more convincing than if you base all of your analysis on a single probe. So we want to find stuff like this. And kind of what's hard about this is that it's not so hard when the array looks like this. We have a lot of probes, we can do it. It gets harder and harder the fewer probes you have in each kind of probe cluster. And we are not completely done with like tuning this, but we think it helps a lot. So what we do is slightly different from what Tiffany talked about before the break. If I understood her presentation correctly, and I've not read the paper, I've not looked at the software, I've just heard her like 3 minutes talk about it, she essentially starts by identifying single CBGs that are differentially methylated using a single CBG test. And then she looks for clusters of significant CBGs. So what we do is we start off with some estimate of the difference between the two things that are based on a single CBG measurement. So this down here is the difference on the M scale between the group means up here. And then we smooth that data, and you can argue, well, is this like a great smoothing? And we can talk about that later. But we smooth the data, and we summarize this entire, and then we kind of take a threshold here, and we say, oh, there's a lot of probes where the smoothed level is like above our threshold. And then this is a putative DMR. So we haven't really assigned significance yet. We've just looked for potential DMRs that are consisting of probes that has a reasonable effect size close together. Then we evaluate the significance of this by doing a permutation testing. So we evaluate the significance of the DMR as a whole and not based on its individual CBGs. And that's sort of a major difference, I would say, compared to what Tiffany did. So we start with a list of candidate regions, we permute the sample labels, we rerun the procedure, and then we see how often we get a better region using some summary statistics. But we compare across the entire genome. This is also different from when people do permutation testing in GWAS, where they tend to their null distribution is based on only the SNP that they're testing. We compare across the entire genome, and thereby we correct for multiple testing. Now, one problem with this, is that most probes are not or loci are not in a candidate region to begin with, and we only get a p-value for the probes that are in the candidate regions to begin with. So we don't have p-values for most of the genome. And, well, that's both good and bad. That means we are kind of reducing, to some extent, if you think about it, our multiple testing burden. And also, when we compare across the genome, we kind of get more yield in our null distribution out of each permutation. So you can do this. And as I said, we're not really done with it. And you get this stuff here out of. Now each point here represents a region and not a probe. So we have 600 regions representing 2,800 probes. We get a somewhat better correlation. The plot looks a lot better. You can argue, is this like too stringent? Is this just different? I'm doing something completely different. It's pretty hard to compare. Is my cutoff just more stringent? But I would say it looks better. We would expect to get a higher correlation with gene expression for what we are doing, specifically because we are studying methylation of promoters across tissues. And we know there's a big difference. There's a huge relationship between methylation and promoters when you do a cross-tissue comparison. It's not true for all comparisons, but it's true for cross tissues. Now, the main problem we have with this stuff is that we don't have very many CBGs in each cluster. But there's another fun thing you can do with the 450K array, which is based on a recent finding we and other people confirmed later about huge, large-scale changes in cancer. This here is from our paper. This is some new data. This is normals. This is cancer. This is a 4 or 8 megabase region. And you have a huge hypermethylation on a sort of a summary level across this entire region. And these large regions we call blocks cover more than half or 2/3 of the genome. And in a recent experiment that is still unpublished, we have kind of done this where we are not studying cancer, we're kind of studying cancer, but we are studying a model system where we EBV transform B cells. EBV, or Epstein-Barr virus, is what we're using to immortalize cell lines, but it's also an oncogenic virus, so we are kind of causing cancer. And we can show that this step here is directly related to the EBV transformation. These large blocks of hypermethylation means that the marginal distribution, this is from whole genome by sulfide sequencing, by the way. The marginal distribution of your signal in the normals and in the cancer's genome-wide are completely different. And this is one of the issues why you want to watch out with just blindly doing quantile normalization if you're doing cancer. It may be okay in an EWAS type study, but in cancer you definitely want to be a little bit careful. So we thought, well, can we do this on the 450K array data? Because now we have a lot of probes. Our issue is, again, the probes are not evenly spaced. Some regions of the genome are overrepresented, blah, blah, blah. But this is in colon cancer. This is data from our Nature Genetics paper. This is data from 450K using some publicly available colon and normal cancer. And I mean, it's not like spot on, right? I mean, and you should watch out. You have a big thing here, but that's just because you have no probes in here, right? It's easy to get a big difference if you have a big difference here and they're like nearby. But still, it's kind of interesting that this dip down here, you kind of have here, and you have a pretty big difference here that sort of somewhat seems to be capitulated here. So we actually think that by tweaking this a little bit, we can perhaps not find these large-scale blocks in cancer using a 450K at the same resolution or with the same boundaries, but we can definitely study this phenomena using 450K data. Okay, so that's my talk, and guess we have questions. you could kind of sum up the value here of the line. You can either integrate it, where you don't take into account how many CBGs you have in it. That's probably not great. You can also integrate it where you essentially sum up a contribution from each probe. That's essentially kind of an integration, but it's weighted by how many CBGs you have in it. That seems to work pretty well. You could also say like the biggest difference in the region or whatever. I think that the one we are most happy about right now, but it changes once in a while, is simply summing up kind of the deviation across for the contribution from each CBG. So you are going to be weighted towards having longer regions.
Can I ask you another question?
Yeah.
Did I understand correctly that all of your regions have been defined based on the annotation file before you started?
Yeah. So essentially there's a over limit where you don't really want to group them together and analyze them together. And that's also something we are discussing a little bit. What number should that be? But
another question over here.
Thank you. So thank you for the presentation. First of all, I completely agree with the thresholding based on the effect size, and this is really important, especially for going for second phase validation, because this is dramatically changing the number of results, and this is absolutely required. The question is about the integration between methylation and expression. Actually, how do you define the association of the methylation probe with the gene? Do you just use the Illumina notation? Maybe you just use more sophisticated models where, for example, the genomic region is a bit more complicated. You may have overlapping genes, shared promoters.
I mean, we kind of do it ourselves, but I'm sure that what they have to some extent in the Illumina annotation is fine. I mean, we just say we take all transcription start sites in the genome, and I think we filled out the ones that overlap, but it's only the transcription start sites. It's not the entire gene. And then we just say, is there a probe in that gene? And that, if a gene has multiple transcription start sites corresponding to multiple transcripts, who knows if this particular transcript is expressed? It's like not necessarily great. I mean, it could be, it definitely could be wrong. Some of the bad points we have on the point here, some of the stuff here, I mean, that might be that we are measuring the wrong transcription start site or the wrong place, but that's kind of how we do it. And for this comparison here, we are really only focusing on the 2 kb around the promoter. There is some evidence that methylation in the gene body is associated, and I think that's a fascinating biological question. But I think if you want to like benchmark your algorithm, I think you really have to stick to some observations that have been pretty well supported, I would say, in the literature. And I would say across tissue comparisons, methylation in promoters, that is pretty heavily associated with gene expression.
Okay, one more question. Hi, thank you for an excellent talk. Just from your expertise, would you trust a specific gene analysis from the 450K?
I mean, if I had a disease and I had my favorite gene, I think you should start by looking at that, right, and see if you can make some sense out of it. I mean, what are you specifically thinking about? I mean, of course you can profile a single gene using the 450K.
Using the same cutoffs, Bonferroni and.
I don't really trust the single CBG analysis, but I mean, if you only have a single CBG in your gene and it's really different, I would say that at least is interesting and should be followed up with some further validation using a better, perhaps more comprehensive assay. I think that we are not there where we can really talk about methylation differences in a single CBG being causally functionally related in any way.
All right, thank you.